insect identification keys for me. Yes, I have. What insect do you have? It will be the right question. If you have a certain insect of interest, I think the this forum, this Global Entomologist Network, can help in a great way. I have found an insect in tomato field. This is from Saru. Sorry, the previous question was I don't want to pronounce his name wrong. So I will say Basemon, the last name only I will say. So probably the first name is Jax, if I'm not wrong. I don't know. I'm sorry if I'm saying it wrong.
Yeah.
Next question is from Saru, which is I have found an insect in tomato field which looks almost like Nesidiocorus tenuis, which is a natural enemy of tomato leaf miner. Can you tell me what are the criteria to ensure it is the same suspected insect? All right. So my question would be before you say that it almost look like that particular bio. I will say that this is the Nesidio chorus will be the biocontrol agent. Let me understand this one. So you say it almost looks like. On what criteria you say it almost looks like? Because there is nothing in taxonomy which goes with almost looks like. The over the period of time I have learned in taxonomy, the certain words do not make any sense, like larger, smaller, weaker, stronger. These words do not make any sense in taxonomy because taxonomy deals with the distinct characteristics. If the distinct characteristics cannot be identified or cannot be used, it means the taxonomy that you are using is weak. So first of all, I will focus on this word which looks almost like Nesidiocorus tenuis. So there can be a large number of organisms which do look almost alike. So based on being almost look-alike, you cannot identify a certain species. So there is a way that first of all, what you can do, you can see like very crude way, very crude way, while sitting in your place, you can see the distribution of Nesidiocorus tenuis. You can see that if it is distributed, it has been reported from the area that you are working on, in where you are present nowadays. So if that this organism is being reported, that's good. It means there is more chance that the one that you have is the same species or the same genus. The next step would be, let's say if it is not, the next step would be to reach out towards the uh, distinct characteristics to start identification from the higher ranks. And the first thing that you should have in your hand is the patience, not to be in a hurry, because as my supervisor has always said, it is very easy to break your neck in the taxonomy. A single mistake can lead you towards a very big misidentification and a very wrong conclusion. So don't be in a hurry, relax. See the taxonomic or identification or diagnostic keys starting from the higher ranks, starting from the family level, and then go to genus level, and then go to species level. And unless or until you are sure that the one that you have is the same in its morphological characters, you can make this announcement that you have this particular species. If not, there are some variations. There can be some variations because of which the integrated taxonomy comes. The next step would be to have its DNA barcoding and to see what are the genetic makeup of this organism. However, most of the biocontrol agents, if I just generally assume, is that they will be easy to identify because of their significant interest for the businessmen, for the business owners. They are usually trying to rear them and to sell them to the local community. I will go to the question of Sayyid Muhammad Rehan bin Qamar. For all those who have appreciated the presentation, I thank you in between. Thank you so much for your appreciation. What latest techniques we are using to identify minute insects through barcoding? Okay. Okay. For the barcoding, I think the two techniques that I know is through the RFLP method and the normal PCR sequencing. One thing that you have to make sure that the genes that you are using, the primers that you are using for the DNA barcoding, they are of larger base pair length. This is my personal experience that if the base pair lengths are smaller, the chances of heterogeneity or the base pair, what they call substitution, are more. So if you have a smaller segment of the DNA, uh, the chances to get a strong conclusion will be less. So try to use the primers or the uh, or the genes which are targeting larger segment of the, either this is COX-1, whether it is from the ribosomal or nuclear DNA that you are targeting, whatever, but it should be for a larger segment of the DNA. It will help you to reach a better conclusion. If I have understood the question correctly, and I hope that I have answered it. If it is not, then I apologize for that.
Excuse me,
Uh,
Dr. Jawad.
yes, please.
Almost time is over, but
Okay.
with the permission of our respected participants, I want to continue this session because I want you to answer all the questions because the lecture is very informative, important for especially those who are dealing with taxonomic research and also those who want to start their master or PhD, especially in the entomological science. But I just request try to answer briefly. So we will take all the questions. Then I have some questions and I want to discuss them in a different way. Thank you.
Yes, for sure, Dr. Sakib. No problem. I will just try to briefly uh, answer every question. I will go to the next one, which is SA. Do you think molecular identification is a must to use tool following classification identification procedures? I disagree with the. No, first of all, I let me make it clear. I should have done it before. All the answers are based on my experiences, and this is my opinion. This is not a fact. So everyone has a right to disagree, which is perfectly fine. I'm just giving my opinion based on my experiences and my beliefs. Also, I believe on the things which I am saying. So if I think if it is a must to use, no, I do not think it's a must to use. It is a tool which is used for the confirmation of identity. It is not a must to use. The traditional or classical taxonomy still rules. It has its own significance, and no one should bypass it. So I would not say must to use, but I say yes, it is a good tool to use. Obviously, after you have everything done in the classical identification procedures. And then I have a question from Victor that I wish to ask if there are fully funded trainings for taxonomy, either online or offline. How can I access it? I am sorry, I may not have the exact answer for this one. I was, as I mentioned, I was trying to find those jobs which I saw previously on the LinkedIn profile, in particular, going with the job posting for the curators in the museum. I didn't find it, and I hope that you can find them online. This is the easiest way to get it. But I'm sorry, I may not have a definitive answer
Yeah,
for your question.
Dr. Jawad, inshallah, at our forum at the Gen, inshallah, we will arrange a workshop for the taxonomy, inshallah, how to use taxonomy as a science in their research, just keeping in view the principles of the taxonomy. So, inshallah, we will arrange a workshop by inviting different speakers from different groups of insects or life, inshallah. Thank you.
Yeah, thank you so much, Dr. Sakib. Yes, I would suggest everyone to stay connected with the group on the WhatsApp or on the LinkedIn or other social media platforms, so that the information that you are looking for will appear over there. So next question will be from Miss Mary Barkworth. I would say the question, which is, please talk about the value of collections sharing their specimen information via GBIF. Okay, I have heard of it. Global Biodiversity Information Facility. I have no enough experience to talk about this particular institution or forum. However, I will say the value of collections, sharing their specimen information. Mm, I would say if you can be a little bit, can we have this question in a rephrased way? If Miss Mary Barkwood can ask this question in another way so that I can understand and answer it properly. Thank you. Okay. The question would be from Sushank, which is how to mitigate the virus carrying vectors, how to identify if the plant is infected from virus or not. Okay. Um, I may not have understood this question properly because the second part, which goes with the how to identify if the plant is infected from the virus or not, is the laboratory assay. This is what I can say. If you want to differentiate between the stress the plant is going through, environmental stress, or if the symptoms are because of the virus, the I think bioassay in the assays in the laboratory can help. In the other way, the symptoms can be confusing as far as I know. The first part, which is how to mitigate the virus carrying vectors. Well, for the virus carrying vectors, there are some genetic studies being done for the I think for the Anopheles and for the Aedes, where they were first of all, Anopheles is not the virus carrying. I will correct myself. But I know I have read a study about the Anopheles and the Aedes that there are genetic studies where the carrying capacity of the vector is reduced. So how to mitigate the virus carrying vectors, in particularly virus. I remember one of the conferences I was with my master's supervisor, Professor Dr. Wasim Akram, and the one of the important point they mentioned in the conference was that the problem with the Aedes mosquito or the dengue virus is that a single mosquito once infected, it remains infected its whole life. So means that because in the Anopheles, in the malaria, half of the Plasmodium life cycle is in the host. So it means the chances of transferring, if the Anopheles is in Anopheles and the Aedes are in the same room, the chances of per people in that room getting dengue will be more as compared to the malaria. So mitigating the virus carrying vector would be to first of all reduce the spreadness or the reduce the what we say spread of those uh, viruses in the community through the mosquito bite. So the first step would be to protect yourself as the I forgot the word. It's not defense, but the prevention. So you are preventing. So if you are talking about in particularly plant viruses, so plant viruses, I think again goes with the some genetic studies. There are reports in which the capacity of the arthropod is reduced through some genetic mechanism. This is as I know right now. Okay, the question from Muhammad Sabtain Arshad. Okay, sorry, the above one. I'm sorry, brother. I will say A. I don't know how to pronounce it. I don't want to pronounce it wrong. So the question is, please, when do we use genus SP and when we use genus SPP? Oh, this is for singular and plural. When you have a single species, you use SP. When you have multiple species, you use SPP. And this is in the case of absence of identification. So if you do not have, if you have two, let's understand from this scenario that you have one species from a genus, you will write it SP because you don't know the species name. But if you have two species from the same genus, two different species from the same genus, or more than two species from the same genus, you will write SPP because still you are sure of the genus, but you don't know how many species, what are the names of those species. You don't have proper identification. So this is the problem of singular and plural. Mohammad Sabtain Arshad asks, can misidentification of species affect crop protection strategies? How it cannot. Misidentification of species, I assume this is about the pest species, affect crop protection strategies. Yes, it can. Different species will have different resistance development if we develop a certain scenario where you are using pesticides for the pest management. And in the environment, the pest has a resistance. A certain pest, which is actually crop attacking your crop, is has developed resistance, but you have misidentified it, and you are continuously spraying in the field, and that pest is not dying because you didn't know that this pest has already developed the resistance because of misidentification. Other example in the biocontrol will be because there are two or three different categories of the predators. So going with the specialist predators, which are specialists, which are actively seeking the pest species. So if you have misidentified the pest species, you are most likely going to select a wrong predator. Same goes for the parasitoid. The parasitoids are either stage specific or species specific. So if you have misidentified your pest, you are going to use a wrong parasitoid, which is not going to give any control. So obviously, the misidentification of the pest species can affect can affect crop protection strategies, and it goes both ways. So if you have misidentified a biocontrol agent which does not have any potential to control the pest, still the result will be the same. Miss Mary Barkworth. Oh, you have answered the question. Thank you so much. Yeah, this is I approve this answer, which genus species, one species in genus SPP, two or more. Yeah, this is exactly right. Okay, Iman Rad says my thesis will about genetic in insects. I need information about it. I collect information. Oh, I don't know if this is a question or I don't know. And thank you, Miss Mary Barkwood, for repeating your question. I will try to read it now, and if I have an answer, I will be glad to reply for it. So the question is, GBIF shares specimen data, and that makes it a good source of distribution data. How good the data from GBIF are depends in the accuracy of the identification and how many collections from an area are sharing their data. Okay, I think this is a comment. This is not a question, if I understand it correctly. However, I had the experience with one of the. Thank you, Miss Mary, for confirming. Uh, I have the experience with the other databases where the journals, because I'm from ecology, from the mites specifically, so they ask us to submit the data that we have. I think this is public databases from Zenodo in particular. I think GBIF is kind of like this. I have gotten information from the GBIF quite a multiple times, but I didn't had the experience with them. So what I understand the GBIF would be that the information about the species, its distribution, anything that is related for the species will be uploaded for the use of community. And this is a good way. This is a good way, and it is I think being adopted by those institutions which are dealing with the genetic data like the board or the NCBI. They are also, they do have the genetic sequence data, but bold is more advanced because it also takes the images of the specimen with which the sequences are attached and its distribution also. The bold is rather more organized than NCBI because NCBI only have the genetic sequences. So I think this is a good practice. I would just add my comment here that this is a good practice and this is good thing that the information goes around the world because the pests of quarantine significance are also over there. Pests of endangered species. Yes, exactly. You are right. It should be promoted. It should be promoted, and I agree with that. Uh, that the for the quarantine significance, I have seen those endangered or extinct species data over there. So this should be shared for the people who are dealing with the taxonomy of larger groups. So I am in favor of this. Yeah, it should be shared once it is published, so the and it should be available for the public to be used. I don't see any comments or questions. Okay, can we get yes, yes. This is not my place to answer for the
Thank you.
certificate.
Thank you very much, Dr. Jawad. Mashallah. I'm again very thankful for spending time, your valuable time, to speak for this lecture. And first of all, I uh, just want to again request to our respected participants, just bear us for a few minutes. So I will ask some questions and discuss some important points related with the taxonomy with Dr. Jawad. And firstly, I really salute the supervisor, uh, especially the both of us, as we both graduated from the same lab under the supervision of Dr. Fahad Jabal Alatwi, who is not only a great scientist but also a great taxonomist working with mites in Saudi Arabia in the King Saud University. And interesting thing that I would like to share here that Dr. Fahad, who graduated with master and PhD from some American universities like Kansas and the Ohio State University. But he actually did his research in master on taxonomy, but in PhD on biological control. But when he came to Saudi Arabia, he, mashallah, become the in charge of the ecology laboratory and main research work there on the taxonomic work. And mashallah, since maybe last 15 or 20 years, he's working mainly on the taxonomy, but now some other aspects related with the biological control and some behavior or some IPM sites. But again, I want to say here that entomology is very important. The taxonomy is very important to understand the basic principles of taxonomy for all the entomologists, especially who is working any field of the I can say the entomology, either the applied side or the basic side. So if someone who didn't do his research in master or PhD on taxonomy, it doesn't mean that he could not understand or could not learn the basic or the key concepts of entomology later on. The one example, my supervisor and Dr. Jawad's supervisor, Dr. Fahad. And the second thing that Dr. Jawad, mashallah, he although didn't work on taxonomy, not even his master, not even with his PhD, uh, but mashallah, after his PhD and even during his PhD, because he's also doing a job there as a researcher in King Saud University, he published almost maybe 30 plus papers on the taxonomy. And I think he's now a good researcher in taxonomy as compared to what he specialized in master on medical entomology or some, I can say, some the behavioral or the biological control studies in his PhD research. So the conclusion is here. All the entomologists who are working in any field, either in the quarantine field or the IPM or the biological control or the industry, one should know some basic principles how to deal with any new specimen, either it's a pest or some beneficial or some invasive species like that. So one should try to understand and learn the basic principles of taxonomy. So now coming to the point, uh, I'm a taxonomist because my master and PhD research in uh, taxonomy, but now since maybe last 2 or 3 years or more than since 2017, I'm working on biological control, some behavioral aspects. But interestingly, we learn many things from our lab, and based on those experiences in the past, in my master and PhD, I would want to ask some basic questions that, inshallah, will be the answers will be helpful for understanding the basic concept of taxonomy for all. So, Dr. Jawad, my first question: uh, how to define and differentiate the taxonomy with integrative taxonomy when we in comparison with the tools we are using, like if we just go with the taxonomy itself, what tools comes in our mind, or what the approach comes into our mind, or while when we deal or they talk about the integrative taxonomy, as I believe that I graduated in PhD or my research mainly not only taxonomy but also integrative taxonomy, like I integrated different tools or different approaches like the morphological, molecular, and behavioral approaches to identify or to discuss the taxonomic issues in some important group of spider mites. So please, Dr. Jawad, thank you.
All right, Dr. Sakib, thank you so much, Jazakallah khair, for your appreciation and for your kind words. And yes, the as I mentioned earlier, that I had the privilege to uh, get my degree from this lab, and this lab has given, under the supervision of Dr. Fahad, too much knowledge and has opened many things in concept for us. Uh, I will just repeat your question, just confirm that if I have understood it correctly. So that you are referring to the differences in the tools of classical taxonomy or traditional taxonomy in comparison to the integrated taxonomy. Is this correct?
Yes.
Okay. Well, the if we say this terminology of traditional taxonomy, the tools that come in my mind will be both published and non-published. Because I will not just keep mites in my mind, I will just keep some larger organisms or arthropods, including mites. So first will come as the published tool, which will be the the literature, the books and the chapters and the keys. At least we in our bachelors we have the identification key of orders that we study from the manual of entomology, the laboratory manual of entomology, that have all the characteristics which can easily identify or differentiate the different orders of the class insecta. So the first tool that comes in my mind for any arthropod taxonomy in the classical or traditional way will be the literature. So the person should have a strong literature grip. The one who is working in the taxonomy should have a strong literature grip. The second tool will be considering in the non-published would be the accessories or the things that we use. For larger arthropods should be a good magnifying lens. Smaller arthropods should be stereo microscopes, and further smaller, which are used on slide mounting, will use the compound microscopes with different accessories of differential interference or the confocal or the what we say phase contrast. So again, for the traditional taxonomy, the two types of tools that come in my mind will be published and non-published ones. For the for the molecular oh sorry, for the integrated taxonomy, will directly go to the molecular technique for the identification confirmation. This is what we have learned through the past experiences and have seen the benefit of this belief that the molecular tool is as has been said, it is a tool, and it is for the species confirmation. And that You have, mashallah, two at least three publications which specifically go uh, with the use of molecular technique for the species confirmation, for the identification of the species. So the tool that comes in my mind for the integrated taxonomy will be molecular tools dealing with the DNA extraction kits or the
Well, Dr. Jawad,
centrifuges.
please. So, another question: can we replace the traditional or the conventional taxonomy, the classical taxonomy, with the molecular taxonomy?
Oh, replace. My again, my opinion would be no. We should avoid the terminologies of replacement and the use of must, as one of the brothers asked the question. The traditional taxonomy or the taxonomy dealing with the morphological characters, it has its own significance, and there is no bypassing this method, this step. The integrated taxonomy comes for the confirmation only, for having a further confirmation of what we have achieved through the traditional taxonomy. Even in these scenarios where we have crypsids, or we have sibling species, or we have morphological identical species, or in your study in particular, where the male is required for species identification, even in that, the traditional taxonomy will lead us to a certain conclusion. So that conclusion will be further, uh, you can say, confirmed through the molecular methods. One of the research papers that we had from our lab was the morphological variations in one of the pest population of on the citrus mite. So the when we talk about the replacement, it means when we are not sure that the traditional taxonomy is telling us what, that the populations that we are dealing with has a problem of sibling species, has a problem of crypsis, has a problem of variations, what kind of problem the morphological characters are showing us. In that way, we can find the answer of this question through molecular tools. But if we directly jump and we skip the part of the traditional taxonomy and we directly jump to the molecular techniques, we will reach the identification, but we don't know what we are finding. Are we trying to solve the problem of species identification? Are we trying to solve the problem of variations in the morphology? What problem we are trying to solve through the use of molecular? So no, I will disagree if someone says that we should replace the traditional taxonomy with integrated taxonomy. Traditional taxonomy has its own significance and it is always before the uh, integrated taxonomy.
All right. Dr. Jawad, I want to share a personal experience. Maybe 2.5 years earlier, I appeared for a job interview here in Pakistan, and one of the respected committee member, he asked me what was the title of your PhD thesis, and I told them that I worked on integrative taxonomy of spider mites by using the morphological, molecular, and some behavioral studies to identify or confirm the species identity. So he asked me a question, or he passed a comment that a question for you, and I want you to answer. He said that if you did molecular, then why you need morphology? So although I answered and I qualified for the job, but now I want to ask same question from you. How you answer this one? Because the committee member actually, based on his experience, although maybe either I think he didn't have any practical experience with the taxonomy, but he asked that if you did molecular, then why you need morphology? Or in other word, there is no need of morphological studies if you have some molecular tool in your hand. So I want you to answer, please.
Well, Dr. Sakib, you answered and you got the job, so it means you are the best guy to answer. Okay, I will try to give my opinion or my thinking, but will I get the job if I answer correctly?
Inshallah, you already have a
Okay.
job.
Okay. In the honest way, or not in the honest way, sorry, but in the scientific way, let's define why we have integrated taxonomy and why we have traditional taxonomy, which is incomplete without the other. In my experience, or let me put my experience on one side. The integrated taxonomy, in particular, talking about the DNA barcoding technique. Let's create a scenario that if I go outside right now and collect an ant specimen or a moth specimen, it is winters here, so there will be no moth outside. There will be no ant outside actually. But let me find something and I will just bring it and deal with its DNA. What information I can get from its DNA? About its genus? No, I cannot get. About its family? No, I cannot get. What I will do that I will take this DNA, I will blast it on the NCBI, and I will get a list of more than 100 sequences with which my DNA is matching. So I am standing in a forked alley. Where should I go? On the left side or on the right side? I cannot decide. So what I have experienced or what I have understood from my experience, that the right way to follow is the traditional taxonomy. Traditional taxonomy is based on distinct features, based on morphological appearances, which everyone can see, because everyone will not have the access for the DNA, but everyone have the access of with the two eyes. If not with the two eyes, then at least with one pair of glasses, or additionally with the hand lens or a stereo microscope, mostly, if we calculate the percentage. So traditional taxonomy is at least helping me to reach to a distinction of family, to the genus, and to the species. Now I am stuck at the species level, as one of the question was in the comments that they almost look like. So what, just imagine if that respected audience member directly goes for the molecular and does not identify or does not rely on the morphological characters. It The person may not reach to a right conclusion because he does not know what the specimen he is dealing with. See, the DNA sequences, they are not related with the morphological appearances. This is what I think we should understand it very clearly, that the morphological characters, they are not controlled by those DNA sequences that we are obtaining. Those DNA sequences, they are conserved regions in a very easy terminology, and they are taking the information from generation to generation, based on which we can identify them easily. So the traditional taxonomy is helping me to reach a certain conclusion. So now why I need the molecular technique? Because now the traditional taxonomy is putting me in a place where I have to decide. In this scenario where I have two different organisms and they are morphologically same in their appearance. So I want to know that if they are one species or different species. If they are different species, their genetic makeup will be different. If they are one species, their genetic makeup will be same. This problem can be more easily understood in the scenario of the morphs, where they have seasonal morphs, where the one organism, same species, had different body shapes or colors in different seasons, in winter or summer. So if someone occurs to have these two morphs of an organism, so the molecular technique is helping me. So it means that the molecular technique alone is incomplete without the traditional taxonomy. But traditional taxonomy is complete, but confirmation is done through the DNA or through the phylogenetic analysis based on sequences.
Thank you, Dr. Jawad. Another question. I think that we heard many times the word scientific principles. So here I want to say the taxonomic principles. So, what are the taxonomic principles? How to recognize, understand, and apply during some conducting any taxonomic studies? Briefly, we want to understand based on your experience. Thank you.
Okay, if I go with the book definition of the principles of taxonomy, then the principles of taxonomy includes the identification, the systematics, the phylogeny, the nomenclature. So all these terminologies, as under the book of the principles of taxonomy, they come. So if we want to talk about them in particular, so the first step in the taxonomy would be to have a grip on the literature, to know that the organism that you have is belonging to which group. Say we have many researchers, they post the pictures in the group and ask for identification, and most of the respected researchers who are expert either in the Coleoptera or the Lepidoptera or the Hemiptera, they just reach to the conclusion directly very easily because of their vast experience through the literature or through the specimen. So it's the first I think the step in the right way of using the taxonomy is the grip on the literature, and on the through the literature we know that what are the significant characteristics of the organism that we want to identify. So
Sorry, Dr. Jawad, we could not hear you.
so first of all, I will put in the significance of taxonomical study is the grip on the literature and the grip on the morphological characters that we study. The second one is the use of right tools. There are different features which are important in the species level identifications, goes in the Odonata, goes in the Diptera, goes in the Hymenoptera, and specifically goes in the mites also. So if the right tools or the correct tools or proper tools are not used, tools I mean microscopes or the related accessories, if they are not used properly, then the morphological characters can be misidentified. Now, this misidentification should not highlight the need of molecular technique. Now, this should highlight the need of better use of tools. No one should misunderstand that if a misidentification is occurring, is highlighting the need of integrated taxonomy. So it is actually highlighting the need of a patience and keen observation. So in the principles of taxonomy, then we move on forward after the literature and use of proper tools. Then is the understanding of the core concept of the characters. Then here the part comes for systematics, that we need to know that if this specimen that we have is a new species, it is going to be a new record. New record means this is first time reported from this particular region. It is not new species for the globe, but it is first time reported from a certain region. It is a new record, or it's a new genus. I would recommend everyone from their own subject to read at least one paper dealing with the upranking of the specimen. For the mites, there is a recent paper in 2025 published where a new genus, which was identified in 2006, has been raised to the family to the level of family in 2025 because of distinct morphological characters which were not properly identified in the systematic way. So in that particular scenario, the correct the person who first time identified it as a new genus, he was having a trouble or problem facing. He was facing problem based on the morphological characters to put it in the closely related family. But however, he put it tentatively, and where he overlooked a certain character on the leg segments. So in the recent years, when it was reviewed, so that genus was raised to the level of family. So at least in your own groups where you are in the Coleoptera or on the Lepidoptera, at least read one paper where the rank of the genus or a lower rank has been raised to an upper rank. So this will help you to understand systematics in the principles of taxonomy. When you have a strong grip of literature, you have proper tools, then you can evaluate that whether the specimen that I have should be placed in the species level, should be a new genus, or must be a new family. These all decisions are not based on genetic sequences, because genetic sequences, again I will mention, are for the species, and species are the basic unit. But to define the higher ranks and to define the categorization, this is based on this goes with the traditional taxonomy. And then the last one is, if I will put in the principles of taxonomy, is the naming, because there are problems of homonyms, same name of different species are present. In association with that, I will also mention that the characteristics should be carefully identified, because if not, then in the later years, the species can be synonymized with the older species. What I mean that if a species is identified in 2010 and I didn't knew about it because of lack of grip on literature, and I identified a new species and published it in 2025. So later years, someone will come and he will find out through the literature review that what Jawad has published in 2025 is similar to what has already been published in 2010. So Jawad species is a junior synonym. It should be synonymized with the one which is already described. So significance of literature with the use of proper tools, identifying the significance of morphological characters, and then naming them in a proper way so that the homonyms situation is not created, I think would be considered for the principles of taxonomy.
Thank you, Dr. Jawad. This answer actually is very uh, informative, especially for those entomologists who are conducting their research in taxonomy. However, I want for those entomologists who never did research on taxonomy, but they are working some applied side of entomology, either with the IPM, biological control, some pest management, either in the stored grain or the agriculture or medical or quarantine related pest management or the structural or urban pest management. So, what would be easy steps for an entomologist if he observed a new insect in his working environment, and how to deal and what would be the steps to follow to at least not maybe species, or at least go to genus or in some major group, that this insect, either it's some quarantine pest or some important agricultural, medical, or any new species, not new species, but or an insect species which having the economic significance. So this I want you to answer step by step what this step should follow. How to confirm he is not it's a taxonomist, but how he can confirm the taxonomic identification, keeping in view the available resources either regionally or the globally, like the museums or something like that. How he can find a way. Thank you.
Right, that's a first of all, I would suggest the person who has seen the let's create a scenario in the
Yeah,
scenario
please.
where uh, uh, a student or a researcher has seen a arthropod of XYZ classification in its vicinity. So first of all, उसको घबराना नहीं चाहिए. He should not or she should not panic. Uh, Look out for the proper tool for preservation. The first thing comes with the preservation. It is very important when it comes for the taxonomical studies, because wrong handling or wrong preservation method or technique can lead towards the deterioration of the specimen itself. For example, in the butterflies or moth, if the scales are removed, the identification becomes more problematic. Similarly, if they are soft organisms and if they are not properly handled or being captured by the pincers or the forceps, they will be crushed. So the first step would be their preservation. So if you have a preservation proper tool or proper equipment with you, it is great. Usually what we were doing as bachelor students, so carefree and not considering anything in our way. So whatever a plastic water bottle had in our hand, or the plastic bag we had, or whatever we had, we just used it. Even the large leaves we have, we just try to capture the insect uh, more carefully. So if you have a proper tool with you, this is good. If not, then use the available resources, but it should be taken care that the specimen is handled carefully. Because if it has lost, it's because we don't know. Right now, it's an XYZ specimen. We don't know what characteristics it has, which are of significance. So it should be, first of all, handled very carefully. The second thing is that, okay, one thing I will add, there are certain body features which can tell you that this specimen can be venomous or can be poisonous. So this should be kept in mind. It is better not to handle a new specimen which you are not aware of anything with your bare hands. It is better to cover your hand with any clothes that you have around you, or any plastic bag you have around you. So personal care is also very important. It is not desired that you find a new organism while you are in the hospital. So you should be alive, you should be healthy, and you should be in your well state while you are preserving the specimen. After the specimen is it is preserved either in the with the proper tools you have or not. The next step comes, the question comes, what is it? Usually the question is asked if it is beneficial or it is harmful. These are the only two questions that we usually have. There are different approaches to reach to this distinct level. First approach would be that if you can find out more specimen or more individuals which are around you. For example, if you are sitting in an office and the office is nearby have a tree or a shrubby vegetation around you, so it's better to find more specimen, because always more specimen will give you more confidence towards the morphological characters that you are going to use. If there is a single specimen, the characteristics can be of less significance. You are not that much confidence to reach to a certain conclusion. So the first approach would be that if you can find more of them, where are they? If you cannot, let's say in the scenario where you are in your office, but there is no vegetation, there is no way it can come from the window or the door, but it came somehow with someone who just visited your office. So it means you are just dealing with the single specimen. So it is good to see if it is a what kind of mouth parts it has. If it is in general appearance, it is with the phytophagus feeding on the plants. If it is blood sucking, if it is feeding on any if it is parasitizing any bird or any mammal. So these are the basic information. They are very critically important in the taxonomy, because when we deal with the taxonomic descriptions, we put all these informations. So this is the basic investigation that you can do while you have the specimen in your hand. The second thing would be that usually if when we deal with the arthropods, they have hard exoskeleton. So it is, if it is alive, it is going to uh, cause a problem with its either with its body. If especially if it is winged, it will try to fly. If it is not winged, it is crawling. It will keep moving and may lose its legs or antennae, which are of important. So the second thing would be to reach out for a certain method where you can just carefully kill the organism. If you are in a vicinity where you have access to the methanol, that is great. If not, then I think in the I cannot name any other method where the organism can be safely stored. It should be a methanol or the I would not recommend spirit, but it is alcoholic, so yes, can be used for a time being. So the main purpose is that you have killed the organism. But if it is winged like Lepidopteran or the uh, which have soft wings, it would be better to put them with their wings together. I learned this technique in the NIPG while working, that if you have a winged organisms, which wings is very important, so you should make sure that it is not flapping, and you should make a triangular shape fold out of your paper, any paper that you have. Put the Lepidopterus organism or the winged organism in that triangular shape patch, and then staple it. And it will after some time obviously die because of its short life cycle in that paper, and the wings will be preserved. So any kind of wing organism that you have, you can just preserve it in the triangle shape paper that you can make. If someone has experience with the making samosa, that's he is going to be expert in this. So the next step would be to reach out for the literature. See, literature is first and foremost is the necessary part of any study, of any study, either whether it is for the science or it is not for the science. So we cannot deny the significance of the literature. So the first tool that you can get is to deal with the literature. But if you have access with the experts, this is the shortest way. You can show the experts who can identify the specimen. If not possible till species, they can reach till genus level. And furthermore, if you have genus, then the next step would be from the online, that you can see what are those species reported from this genus around your area. uh, Usually reported through online sources or published papers. And through that, you can make an identification that if this organism that you have captured is of what significance, biological control or of the pest significance. So I'm trying not to reach to the new species, just to have a species confirmation that we have. So this is the way, the from the starting from the methods of collection till reaching to the experts, or if the person is expert by himself, he should consult the experts to identify the species that he has captured. I don't know if should I go in details for slide mounting, but there are different mediums for different organisms. In mites, we use Hertz medium in particular for most of the mites specimen mounting. But in the family, for the family Areophidae, for the gall mites, we use Kafer medium. So there are different mediums in which the specimens is preserved. We have Dr. Iftikhar from the museum in the Kingston University. He has used the potassium hydroxide for the thrips preservation or for processing. So yeah, the processing and the mounting is very important, because most of the morphological characters, as I have mentioned again and again, they become the artifact of improper mounting. And those which features which are not usually there, they appear, or those features which are usually there, they disappear, which lead towards the misidentification. And it can be solved with the point that I have mentioned in the start, that if you have more specimen, you have more confidence about the absence or presence of the character. This brings me to my last point, that how to identify the significance of the character. This is what my supervisor have told me, that the number one uh, step is to see the characteristics which are of absence and presence category. This is the important and significantly strong characteristics on which basis you can differentiate. The second one, after the absence and presence of character, would be the position and then the size.